Welcome again to paboardreview.org. I've created this wiki review on the physiology of lipid absorption and distribution. I will tie the concepts Joe shared in the cardiology lipid lecture. The wiki reviews will be a very high level look at the physiology and pathophysiology to help us understand the process and not just brutally memorize facts. Remember, plasma is a poor transporter of cholesterol. So cholesterol and its components are therefore transported around the body in structures called lipoproteins, which are a combination of lipofats and proteins. The ratio of fats to proteins determines the structure and therefore the function of each type of lipoprotein. This slide is to give us a visual of what we'll be talking about during the lecture. So we're going to look at the percentage of fat to the percentage of protein. Micelle are fat droplets that have been broken down via bile salts, or another name for that is emulsified, into small absorbable units. So during the lecture, when we talk about the lipoproteins, we'll be talking about chylomicrons, VLDL, LDL, HDL, and you can see the orange represents the cholesterol or phospholipids, the gray represents the protein component, and the blue represents the phospholipid. The structure all the way to the right is a structure that can contain any of these components but doesn't represent a particular lipoprotein. We're going to talk about a few other physiological processes at a very high level to understand where some of these pharmacological interventions work. And we're going to stay very high level. And so remember, glucose, fats, proteins, vitamins, and other important nutrients are broken down and absorbed in the duodenum. Enterocytes on the intestinal side use sodium glucose transporter to take in glucose. Glucose is then transported by way of hepatic portal vein to the liver. We're going to return to the liver and its importance in a minute. Dietary fats are also absorbed in the duodenum. Lipids must be emulsified, or its surface area must be reduced with bile salts in order to be absorbed. They're made into what we call micelle structure. Keep this in the back of your mind. In order to be absorbed into intestinal cells. Micelles are absorbed as fatty acids, monoglycerides, cholesterol, and proteins called apoproteins. These components are all packaged together for transport via the lymphatic system in structures known as chylomicrons. And remember the slide previous to this, we said a lipoprotein is a percentage of cholesterol to protein ratio. So this chylomicron is then dumped into the venous system via the left subclavian vein. The chylomicron then circulates throughout the whole body before it reaches the liver, delivering triglycerides, phospholipids, the components that the cells need. The remaining remnant of the chylomicron will arrive at the liver, binding to LDL receptors and brought into the hepatic cell for processing. We'll talk about the chylomicron a little bit later with the glucose. So, bile acid sequestrians. They work by inhibiting the emulsification of the lipid droplets at the intestinal side through the bile salt. So they work in the GI tract. You can use them with people with liver disease because they're working on this particular absorption process. Now you can see where it is. Now we're going to return to the liver with glucose. So glucose, through the process of glycolysis, we get pyruvate. Pyruvate can be moved into the mitochondria of the liver cell and become acetyl-CoA. Acetyl-CoA is brought outside the mitochondria and can be converted into cholesterol. So this is another important step. So it's converted to cholesterol through an enzyme called HMG CoA reductase. So this is where the statin drugs work. They specifically target the HMG CoA reductase enzyme, thus limiting the synthesis of cholesterol. Outside the mitochondria, acetyl-CoA can also be made into fatty acids. So glucose, when it is brought into the cell, can also through glycolysis be made into glycerol. Glycerol with fatty acid makes monoglyceride, and if you add more fatty acids, makes triglycerides. Now, we're gonna return to where the chylomicron is brought into the liver through the LDL receptors. So we have fatty acids, monoglycerides, cholesterol, apoproteins. That, along with the monoglyceride, we go into the Golgi apparatus, and that's where the lipoproteins are made. Okay? The liver only makes HDL and VLDL lipoproteins. The HDL are empty. Joe calls them trash trucks. They pick up excess cholesterol and bring them back to the liver. But we'll talk about what these do in a minute. So, the HDL, or the trash trucks, enter the bloodstream. The excess cholesterol in tissue will be picked up by the HDL, and the full HDL will return to the liver and enter via scavenger receptors. The full HDL is endosized, and the HDL that is full now could either be processed again through the Golgi apparatus or excreted via bile, depending on how much cholesterol is needed. Remember, the liver produces HDL and VLDL. VLDL will transport fatty acids and other components to tissues for storage and other needs. VLDL will come across lipase that will liberate the packaged components to tissue for their use. VLDL will stay in the bloodstream and become IDL, which is intermediate density lipoprotein, which is basically empty. IDL then can be converted to LDL to transport cholesterol to other tissues. Now, LDL has the highest cholesterol to protein ratio, more than any other lipoprotein. When we talk about coronary artery disease, the LDL lipoprotein is the #1 target, and now we can see why. The cholesterol is used in various tissues in very many different ways. It's very important. For example, to make hormones, testosterone, estrogen, maintain cell membrane integrity. It's when we have an excess that we run into problems such as coronary artery disease. After LDL gives up cholesterol, it can return to the liver by binding to the LDL receptor, and the LDL can be recycled in the Golgi apparatus to make more lipoproteins, or the excess cholesterol gets excreted in the bile. Now, when we look at fibrates like gemfibrozole inhibits peripheral lipolysis, so that's where it works. It also decreases hepatic free fatty acid extraction. It inhibits synthesis and increases clearance of VLDL carrier apoproteins, but the mechanism of the HDL increase is unknown. I want to give some background information on the cholesterol notes so that we can understand the complexity of it, but also to help us understand the process. So basically, Joe's notes take what's called a ATP, which is the Adult Treatment Panel 3, which is part of the National Cholesterol Education Expert Panel on high blood cholesterol in adults. So this is a 280-page, very technical document, and Joe's broken it down into its very most basic components. But I thought this background would help, especially for students and those who are in specialties like myself who don't have a lot of exposure or real-world experience to this. So a couple of the components I wanted to look at is, now we can understand why LDL is the major atherogenic lipoprotein that is targeted, because it has the highest cholesterol to protein ratio, and it's developing this coronary heart disease is developing earlier in life, and we're learning that we need to be more proactive and prevent its progression or from it even starting. But I thought also looking at the stages of the coronary heart disease, in the first stage we have fatty streaks. Those involve macrophages, and that's derived from the LDL cholesterol. Second stage, fibrous plaque, scar tissue, lipid-rich core. And then the third stage, development of unstable plaques that are prone to rupture, formation of lumen thrombosis, plaque ruptures or erosions, and this is what's responsible for most acute coronary syndromes such as MIs, unstable angina, coronary death. To see the rest of the video and Joe's lecture on cardiology and lipids, please join us at paboardreview.org.